It's just an absolute treat. And so my name is Ryan Kelly. I'm the director of the eDNA Collaborative, and I have two children leaving for school this morning in the next 5 minutes. So this is the most important 5 minutes of the day. So if you hear chaos, that's what's going on in the background. And so about 18 months ago, we started the eDNA Collaborative, and the goal is to take environmental DNA methods and move them out of theory and into practice around the world. And we thought, how can we do that? What are the ways, what are the sort of theories of change in the world that we could try and work along so that we create the biggest effect that we can? And we've worked along all of these lines at the same time, simultaneously, where we do core research and try and make eDNA more effective, better, cheaper, faster, move it into sort of official channels as much as possible. We've been able to award a couple of small pilot grants to get people started with their projects. We've had really fantastic experiences with visiting scholars, including Nuo, who's with us today, come and visit us in Seattle and work with us in what's a very two-way exchange of information. We have learned a lot, and I hope that they have learned the same amount, and it's really been a joy. But what has really become core to what we've been doing is we call microgrants, and all of you are recipients of a microgrant. And so small bits of funding for one thing or another to try and reduce the barriers to entry into working with environmental DNA. And eDNA can be expensive and can have these sort of substantial upfront costs, and some of that is cost in materials, some of that is expertise. And what we're trying to do is just reduce those barriers to entry so that we have more people participating worldwide, in all kinds of research areas. And it's been really fantastic. We now have more than 100 microgrants awarded to people in over 40 countries. And we realized that now the next step is to try and take those people. We know all of you at some level, we know your names, but you don't know each other. And so what we'd like to do is stitch micrograntees into a network so that they can support one another and reach out when you need help. Now you have 100 people to help. And that would be the best case scenario, where you have a support network to rely on. These are potential collaborators and potential friends. So that's the goal today, is to get people talking to one another, connected to one another. For those of you comfortable with sharing your contact information, that's why we're collecting contact information. We're not selling it. And in fact, we're not selling anything. We're just trying to help you meet each other and do cool science in a more, in as effective a way as possible. So that's what today is about. It's about creating community and hearing from you what you're up to, and then having you connect with each other. And that's our goal. And I think with that, we can hand it over and introduce the rest of the team here. As I said, that's me, obviously, on the left, and I'll turn it over to Kara.
Thanks, Ryan. Hi, everybody. My name is Kara Sucher. I know I've emailed with probably everybody on here at some point, so that's really great that I'm not actually get to meet you in person. So I'm very excited for this and hear you talk about all the science I've kind of seen pass by me a little bit. My background is actually in engineering and geochemistry. I did some paleoclimate work before I started working on the logistics and operational side of science. I worked in Antarctica for about 22 years, coordinating some of the research and science we do down there. And then I got very, very lucky and found myself working with Kate and Eiley and Ryan for almost a year and a half now on this eDNA project. And I'm really enjoying it and thrilled to meet you all, and very happy to help facilitate this meeting. So I'm going to pass it on to Eiley now.
Hi, everyone. I'm Eiley Allen. It's so nice to see all your faces, and I'm really looking forward to hearing all about your science. I'm the chief scientist here at the eDNA Collaborative. So I've been doing eDNA for about 10 years now. I mostly focus on fish and marine water, but I'm really curious and interested in all things eDNA. And I am just thrilled to be here. So We're excited to get your talks going. I think Kate mentioned, but I'll introduce the first speaker, and then after that, we'll have you introduce yourselves as your slides come up. And just a reminder that the order of presentations is on that Google Doc. So I think, and I will also be the timer, so I will give you the 3-minute warning in the chat, and then if you're going over, I will just chime in, unmute myself and chime in. So thanks so much for being here, and we'll start with Aaron Sofer.
Hello, everyone. Can you hear me? Good. Okay, so first of all, I'm Aaron Sofer from Tel Aviv University, Israel. And just for context, my research is, I perform my research in the Gulf of Aqaba, which is the northeast corner of the Red Sea in the southern coast of Israel. My research has two main goals. First of all, give a biodiversity assessment using environmental DNA of the Gulf and give some functional diversity of the species we can identify using metabarcoding. My focus is on anthozoa corals and echinoids. And my second goal is to try and use environmental DNA metabarcoding to monitor the mesophotic reef. For those of you who are not marine biologists, mesophotic reef are the reef between 40 meters to 160 meters. We sampled 3 different points across the Gulf. Each point was sampled in 5 different depths, between 10 to 200 meters. The water samples were filtered, and a week later, the DNA was extracted. Right now, we are preparing the samples for the sequencing, which is very exciting. And as for collaboration, my research group has a lot of knowledge, especially about echinoids, and we can really help with this animal's ecology and how we can use environmental DNA to monitor these really nice animals, and everything related to mesophotic reef. And we're pretty good in both of these fields. Of course, happy to be here. So next slide.
Aruj, I think you're on mute, so we are not hearing you. There you are.
Yeah, I am.
I can't tell if she's frozen.
Aruj, we might come back to you if we don't hear from you soon. We can always come back to you at the end of the presentation. Oh, she's back in. Here we go. Let's see if this will work. She dropped out of the meeting. All right, Aruj, we're going to come back to you at the end, and I'll move on to the next slide. But don't worry, we'll come back after everyone has presented.
I am extremely sorry.
Oh,
Okay.
Oh, Aruj, if you're here, we can go.
No, it's okay. I can continue after you. I am sorry.
No, we're ready for you. Don't worry.
No problem at all. Yeah.
Okay, thank you. So I am Aruj Nasir. I am from the University of Punjab, Pakistan. I have worked on the ectomycorrhizal communities of different trees, mainly on Quercus, as well as Pinus cedrus. Nowadays, I am trying to work on the seasonal impact, especially precipitation, the main driving force to lead the ectomycorrhizal community. So in the if you can click the animation, please. Here is the thick forest of Quercus in Ghana. That is about 26 meters above sea level. level. We have collected the ectomycorrhizal fungi in the form of fruiting bodies, as you can see in figure number 2, and in the form of morphotypes present on the plant roots. Then these ectomycorrhizal fungi will be have been analyzed by different kits and modified seed trap method, in which eDNA has sent me the appendroft micropipettes that help me a lot. After sequencing, the sequence will be aligned, and then they will be analyzed in the form of phylogenetic tree. So my main focus is on fungi, mostly ectomycorrhizal fungi. Side by side, I am also working on lichen diversity. For collaboration, I would like if someone will help me in phylogenetic analysis, especially Bayesian analysis, or Mr. Beast. And if I can help anyone in ectomycorrhizal fungi or arbuscule fungi, I am always here to help. Thank you.
Hi, everyone. Can you hear me? I hope yes.
Yes.
Oh, great. My name is Chiara Claire. It's the same, Manfrin, from the University of Trieste, Italy. I'm a molecular zoologist. I started my research career with transcriptomics, so to study stress, immune responses of invasive crayfish. And through my previous previous studies, I've gained knowledge in designing oligonucleotides, probes, and working with qPCR. About 4 years ago, I moved to eDNA, which is a new branch of research in my department. So we are just starting to study the distribution of target species in our region, mainly in freshwater. So at the moment, I'm focused on fish and crayfish. I've used the DNA barcoding to produce distribution maps. In particular, we are trying to conserve, to protect a species of crayfish which is living in Italy only in a small spot of our region and in no other area of our country. So it needs a lot of protection. And using eDNA is very useful because it's a elusive species. Is very difficult to see, observe specimens in nature. So eDNA is very useful in this context. I'm starting now DNA metabarcoding, so I'm quite a base level, but it's very interesting. I've already done a sequencing on different samples of water collected in my region. And for example, that's why I'm really interested in being here. I found differences in, even if I used, for example, I monitor CO1 as a marker, just using different oligonucleotides, I got different outputs in terms of list of species, for example. So, well, in general, I'm interested in getting to know the DNA community for possible joint projects, and would like to help to expand the use of eDNA in routine environmental management. Next slide. Oh.
Can you hear me clearly?
Yes.
Okay. Hi everyone, I'm Dale Anakal. I'm a PhD candidate from the University of Łódź, Poland, but I'm originally from the Philippines. So my research focuses on the tropical arboreal tiger beetles, so specifically on one genus, Doratus. These genus is commonly found on the lowland forest vegetation. So a quick overview of what are tiger beetles. So as you can see in the figure, tiger beetles are voracious predators and are remarkable not only for their speed, diversity and abundance, but also for their potential role as bioindicators. So many species are known as habitat specialists, so they can found in a narrow habitat and one or two the same ecosystem or habitat, making them highly sensitive to environmental changes and potential extinction. Our goal here is to understand how these beetles coexist and strive in a shared ecosystem. So we hypothesize that morphological differences and resource partitioning through microhabitat and prey preferences may facilitate this coexistence. So all the specimens collected here were collected using entomological hand net and preserving a 96% ethanol. So during this first test, DNA extraction were done for 200 samples using a Kiagen kit. So primers BRBF were used for DNA amplification for CO1 gene. So the next step also involves using an excluding primers designed to amplify the DNA of various range of invertebrates while excluding the beetle's DNA. As for now, I'm just new to this method, and we are just receiving the sequences and will process the sequences. In this study, there were 4 pairs of coexisting beetles, and our findings consistently reveal the very significant differences among coexisting species body size. So one species is consistently displaying larger size than the other when they are coexisting. Additionally, the data suggests that larger species tends to inhabit larger leaves, suggesting a link probably between the body size and their preferred foraging substrate. So utilizing eDNA techniques in this research represents an innovative approach for analyzing diet composition of this beetle group and marks also a significant milestone in advancing our understanding of tiger beetle ecology, especially nowadays, for the crucial conservation efforts in the tropical region where both biodiversity and human impact are prominent. Being relatively new to this method, I'm eager really to understand and gain new skills in bioinformatics through training opportunities, and also to look forward to establish collaborative effort with the other researchers specializing in invertebrate studies who also share the same interest in leveraging eDNA for biomonitoring research. Thank you for your attention. Next slide, please.
Hello, do you hear me? Okay, so hello, my name is Ευτυχία Τζαφέστα and I'm a PhD candidate at the University of Salento in Italy. I will present you part of my PhD, which includes the phytoplankton diversity assessment in a Mediterranean coastal lagoon. Coastal lagoons are ecologically significant ecosystems. They provide valuable services and they also serve as important areas for conserving biodiversity. However, they're highly sensitive to both environmental changes driven by climate and human activities. One way for assessing the health of these ecosystems is by studying phytoplankton, which displays an extraordinary diversity of species in these transitional waters. The study of phytoplankton in coastal lagoons using advanced molecular techniques, such as the eDNA metabarcoding, is relatively new, which means we have limited available data. And in addition to that, applying eDNA metabarcoding to a coastal lagoon can be challenging because of the high content in humic substances in these environments. In this study, we gathered water samples from 7 different locations within a lagoon in the southeast of Italy. Specifically, we collected 3 replicates of 1 liter of water from each station. The DNA extraction was conducted using the DNA EasyPar water kit, and the PCR amplification targeted a 390 base pair fragment of the V4 region of the 18S gene. After taxonomic assignment, only the operational taxonomic units that were identified as phytoplankton were kept, resulting in a total of 2,293 phytoplankton O2 clusters. And you can see in the figure how these O2 clusters are distributed among the stations at the phylum level. You can see a slight diversity in the phytoplankton community structure among the stations, which could be due to environmental factors such as the water chemistry, the physical characteristics of the water column, or the nutrients availability. And I say this because, especially in transitional water ecosystems, environmental heterogeneity is the main factor that influences the phytoplankton community structure. Regarding my research journey, I would be very interested in applying eDNA in marine environment and also explore its potential for species-specific detection. Thank you. Next slide.
So Hello, my name is Juan Diego Duques. I'm a PhD candidate in agricultural science at Universidad Nacional de Colombia, and today I'm excited to share with you some results of my research on the effect of climatic seasons on the soil microbiome in forests and paramos of Colombia. In recent decades, the planet has undergone significant changes in climate patterns that have caused changes in the functionality of different ecosystems like forests and paramos. And in Colombia, these ecosystems cover around 52% of the country's surface, making them the most extensive ecosystems. One way to understand how climate change affects this ecosystem is by analyzing the effect of climatic seasons on the composition and diversity of the soil microbiome, considering that this microbiome is crucial in soil and ecosystems health. So this is why we are using metabarcoding in 140 soil samples from 3 different ecosystems: tropical dry forest, Andean forest, and paramo. We amplified and sequenced the 16S gene for bacteria and the ITS for fungi. So far, we have analyzed the microbiome during the dry seasons, and we are currently processing the data from the rainy seasons. As you can see in the graph, we have found that as the altitudinal gradient increase, bacterial phyla like Proteobacteria and Aciobacteria, as well as the fungal phyla Ascomycota, increase in abundance. Furthermore, we have identified a close relationship between pH, organic matter, and the changes in the abundance and diversity in the microbiome. At a functional level, we have discovered that more than 60% of the bacterial functions are related to carbon fixation, and one of the major challenges of the research has been the access to the sampling areas and the return to the laboratory, which could take more than 3 days after the field work is complete. So we are looking for grant opportunities and additional funding to set up a suite case lab. Also, I believe there are exciting opportunities for collaboration to expand these studies to different forests around the world. So we are looking forward to welcoming anyone interested in the subject. Thanks, and next slide.
Hello, I'm Kariama and I work at the University of Strasbourg. I'm a PhD student and I am studying ponds. So my project is mainly about ponds that were created for Emys orbicularis, the European pond turtle. And so I use eDNA, one to detect the presence of amphibians and turtles, but also I'm doing a comparison with normal macrophytes and macroinvertebrates surveys and eDNA methods, and to make a full to see their communities as well. And what we did, we took pond samples, 1 liter, and we filtered them with Sterivex filters. I cannot recommend them because afterwards in the lab it's very impractical. And then we put them in Longmire's buffer. We did an extraction, and at the moment we're still doing the PCRs, and we'll send it for sequencing. So I don't have the results yet. Yes, I'm looking forward to if people are also working on standing waters, and of course also for the bioinformatics and especially multi-locus metabarcoding, which we're trying for the plants, because it's a bit hard to do them with only one region. So yeah, and if people have experience with the regions or the specific target groups, then yeah, I'm happy to exchange. Thank you. Next slide.
Hi, everybody. I'm Luciano Pastorelli, a recent graduate student, University in Peru. And I'm looking to evaluate eDNA metabarcoding as a tool for fish and benthic vertebrate diversity monitoring in central coast of Peru. So these organisms are constantly monitored within these large marine ports that provides an habitat and an artificial protection. So traditional methods are usually also invasive and require a lot of time and effort in field, and also the turbid water of the region does not always allow us to apply some visual surveys for monitoring. So in this regard, the eDNA is being evaluated as a monitoring tool. And within this humble ecosystem is one of the most productive systems worldwide. So it's important to know how does it perform. So we are evaluating this by performing metabarcoding using 12S and CO1 primers in water and sediment samples. The processing will be done by Joanna Ventures and also bioinformatics, but we are currently developing a custom pipeline and will perform also data analysis with R, and also looking to develop a Shiny platform in R for eDNA visualization and data analysis. Well, results are currently in process, and as for future collaboration, I'm encouraging to develop studies on coastal fish ecology and genetics with eDNA within this system that these organisms are fully studied. There are endangered species also. By myself, I'm looking also for training courses as lab or bioinformatics, but especially on laboratory processing, because I have not done this yet, but I'm willing to, and I'm also open to collaborate with projects and develop new ones. Thank you very much. Next slide.
Hi, everyone. It's a great pleasure to be here today. I'm Maja Mucko. I'm a postdoc at the University of Zagreb, Faculty of Science, and I'm a part of the Laboratory for Biological Oceanography. And currently, I'm involved in this project called Island that is primarily searching to evidence on physical forcing changing the biology in the water column. So we are working on this unique island of Lastovo that has a unique physical forcing called internal trapped waves. And these internal trapped waves are just like clockwise waves that are causing the oscillation in thermocline, and they are changing the community, and they're changing the net primary production. They are giving the nutrient fluxes from the bottom to the surface layer, euphotic layers. And we are currently just researching on dense sampling events during the stratification period in summer. And my job is actually to go and see how the community changes. So from the perspective of bacteria and perspective of eukaryotes, I'm doing metabarcoding on both 16S and 18S genes. And basically, I'm trying to find the evidence of this physical forcing. And our preliminary results, I'm very happy to have proven that bacteria are really, really good in responding to this slight changes in daily changes in physical forcings. And our community really shifts towards some other bacteria phyla. And I've just started to do some Faprotax analysis, trying to get the ecological function of this bacteria. But what I'm primarily focused on is to introduce the metagenomics in my lab, because I've been doing metabarcoding now for about 4 and 5 years, and I would like to have a next step to metagenomics to try to answer these ecological questions and metabolic questions in connection of bacteria to phytoplankton and to zooplankton, so the whole community in the water column. So any collaboration of that type or experts in metagenomics on water column will be really, really great contact for me. Thank you. Next slide.
Hi everyone, I'm Michelle Taylor and I'm a PhD student from the University of Aberdeen in Scotland. So my PhD project is looking at the diversity and function of coral rubble beds. For those that aren't aware, coral rubble is the sort of dead coral skeletons that are left behind after coral dies. It can be natural through storms and through bioerosion, but more and more it's happening because of anthropogenic effects. Especially bleaching and subsequent coral mortality. I sort of chose to study this because unfortunately we are past the point of no return in trying to save all of our coral. So understanding that these sort of leftover skeleton beds can be important, I think is really important to know about before we've increased them. What I've done is just got back from my second field season and have been using a number of different methods to kind of try and work out what lives in there and hopefully figure out why and how they connect to other marine ecosystems. So I've been collecting physical specimens. My house is currently full of worms as I go through trying to image them. I have stationary videos from all of my dives, and the specimens are going to be sent for genetic analysis. And I've also collected sort of from each dive 3 eDNA water samples as well that are hopefully going to be sent for some metabarcoding. My results so far, I haven't done any of my eDNA work yet, but physically we found all major marine phyla, which is pretty exciting. We weren't sure that there'd be such diversity in there, but there is. And I've sort of just starting to get organized to get back into the lab now that I'm back in Scotland with all of my samples. My future collaborations and needs has changed drastically since I wrote this like 2 weeks ago, because I have just found out that both of my supervisors are leaving. So at the moment, we have nobody at my university who has any eDNA knowledge, which means that although I have sort of 100 eDNA filters sitting in the freezer, they might just potentially get left sat in the freezer unless I can find sort of someone that is really interested in providing some support with that. Before, my concerns were that we just don't really have the inventory of coral rubble beds barcodes to use as a reference, and especially kind of expanding out into global regions. I'm building the database with my specimens that I've collected before I run the eDNA. Now I'm just hoping that I can do something with these filters, so that's changed massively. Next slide.
Hi, I am Ikram. I'm from Hasanuddin University, and I am an incoming PhD student at the Museum for Naturkunde in Berlin, and which brings my project titles. My PhD project title is actually Species Diversity, Distribution, and Community Assembly of Freshwater Macrofauna of Sulawesi. So I will be doing a sort of comparative approach, so doing sort of a biodiversity exploration to find out what diversity, what kind of freshwater biodiversity in Sulawesi, which is well known for its high endemicity, but at the same time, actually there are a lot of pressure that's going on. So the first thing that I will be doing, actually doing first biodiversity exploration, collecting more data, collecting more DNA barcoding, and to build a good reference. And then eventually I can use eDNA metabarcoding at the end. So, which I don't really describe the methods here. It's just like a general things that I will be doing. So first, more exploring various ecosystems such as subterranean ecosystem, ancient lakes system here in Sulawesi Island, and also remote upstream, which many of them have not been yet explored. So it's something that I will be doing in my PhD. But anyway, for the time being, I had a small funding from the university to conduct small project. My very first project of eDNA which I conducted not far from the city where I live in, and yeah, it is the very first time. And then I compared the traditional methods of using gill nets and also using eDNA. And at the end, I actually found more species using eDNA compared to the traditional methods. But there's one interesting point from my result using eDNA. Actually, there is one species that I see directly in the water, but then it's not detected in the using eDNA. So the species here, rainbow fish, so it's not detected using eDNA, but it's actually there. I catch it, I see it, but I don't know what's going on. Maybe the primer that I use, because I use the my fish, which is largely used for seawater. I'm not sure if the freshwater can also work for this fish. So maybe something that I need to read for. And yeah, this is maybe the things that I'm doing. What I've been doing also here in Sulawesi, because it is a new thing, eDNA is new things for many people here. So I try to involve more students, bachelor students, to get involved in the project that I'm doing. So getting them to the field, filtering the water, and like letting them know what is eDNA actually. So yeah, something that I'm doing, and then this is my very first stepping stone, my very first project of eDNA, and hopefully there will be more collaboration with people from the eDNA collaborative. So yeah, hopefully I can explore more about this island. And I'm really sorry. Lastly, I'm really sorry because I'm late. I'm in the field now doing my sampling campaign for my PhD, so collecting more invertebrates and also fish in freshwater ecosystem here in Sulawesi. Yeah, that's from.
Hi everyone, my name is Watawa Kofino from Ivory Coast. I can define myself as an environmental scientist because I'm working with, let's say, invertebrates and also bacteria, which are really my focus. But today I will just try to present the work we have done in the Bandama River, which is a specific river highly impacted by pollution. And in this study, we only focus on one aspect of eDNA approach. It is metagenomics. Clearly, we start with sampling, and then after that, we have, let's say, a extraction process focused on 12S DNA, RNA as a target for identification. And finally, we have, let's say, a partner in UK named Naturemetrics, who perform all the sequencing analysis for us and then send us the result. Then we have the possibility to, let's say, analyze the results. And then what we have used as, let's say, topics of analysis is the OTU clustering. And clearly, the result we showed with this result is like, we have many species in this, let's say, surface water system. And what is really impressive, what is really encouraging for us is like, we try to give the whole information of the water with only 1, 2, 3 replicates of samples. And this shows that exceptionally, we have, let's say, a kind of variety of species from fish to others compared to mammalia and also amphibia, and particularly some reptilia, which are really in, let's say, in the red list. We would like always also to try to focus on in the next studies. Clearly, what we are doing now is, okay, we have a lab, and then we have the possibility to perform barcoding, metabarcoding, and metagenomics. And for the next studies, we would like in one study to do the both things. It's like, for example, we have the traditional fishing capture we use to have, let's say, a kind of barcoding species to identify, perform metagenomics, but also include in this system the metagenomics, including bacteria, archaea, and also microeukaryotes. It is something which is really important for us. And then in this scope, we really have hope that we will have, let's say, collaboration with people who are interested. I know that there are 2 people who explain 2 things interesting for me. I can be in touch with them because they are trying to work on the sediments or the metagenomics of Legon, something like that. I'm really happy to be here today, and also thank everyone for this great day. and hope that we have a very interesting network to work together. Thank you a lot. Next slide.
Hi everyone, it's great to be here. I'm Sofia Nogueira. I'm based in Lisbon. I work at the Fish Invasion Lab, and currently I'm kind of providing support to several projects, and that could be divided sort of into the diet metabarcoding projects, which we had one studying the diet of the invasive tilapia. And now we have an ongoing project trying to study the diet of the Silurus glanish. And this is it's actually the biggest European fish. And we're studying in the Tagus River. In Portugal, there's 2 rivers where it has spread, and this is one of the locations. And then I'm also involved in the single species detection using eDNA. And last year we had a project trying to detect an endangered fish species. And actually what came out of that project, which was very interesting, was that we were able to detect this species in some locations where it had not been detected for over 25 years. And now we have another project doing qPCR to detect the early signs of the invasive European perch. And yeah, I look forward to see what everyone else is doing. At the moment, I'm looking for a PhD position, not necessarily with eDNA. Right now, I'm also, I wanted to get into phylogenetics, but yeah, it's great to be here, and thank you. Next slide, sorry.
Hi, everyone. My name is Tessa, and I'm a second-year PhD student, currently working on my PhD at the National Museum of Natural Sciences in Madrid, Spain. I'm from the US, but I moved to Spain about 6 years ago to do my master's degree in conservation biology. And then I decided just to stay here and complete my doctorate. And my current research involves the use of eDNA for understanding the impact of invasive species on endangered fish in the Mediterranean coast. Currently, I'm interested in 2 species that are often found living together simultaneously, and that's the endangered Spanish tooth carp and the invasive eastern mosquito fish. Since the arrival of the mosquito fish in Spain, Spanish tooth carp populations have been declining at an alarming rate. And we wonder if this is due to a competition for resources or to predation. So that's why we've decided to carry out a diet study. And we just started this, so we have begun a preliminary study at one sampling site in the south of Spain, where we found both of these species living together. And we sampled 25 individuals per species and collected feces. After giving both of them like a very soft stomach massage, and we collected the feces on a sterile swab. And then we extracted the DNA and sent that off for sequencing. And we did DNA metabarcoding using the 12S primer from MyFish. And following our bioinformatic analyses, we found high amounts of host DNA within each host species, but that's normal because the feces were coming from them. And then we found only 1 sequence read of mosquito fish DNA inside the tooth carp feces and 10 tooth carp feces or 10 tooth carp reads within the mosquito fish feces. And after we did a statistical analysis, we found this to be statistically significant. So we think that it is possible that the mosquito fish is preying on the Spanish tooth carp, whether it be eggs or juveniles or even scales, because mosquito fish are known to be quite aggressive and predatory. But we have many more populations and individuals to sample before we come to any real conclusions. So this is very, very, very preliminary. So for this reason, some collaborations that interest me would be if anyone's done diet studies based on fecal matter, especially for small organisms. And if anyone's worked with blocking primers before, and diet studies using COI for invertebrates to see competition for resources. And any advice in general is welcome. Thank you.
Wow. It's like I can't possibly type fast enough. Like typing as fast as I can and then trying to pay attention to one person at the same time as responding to everyone else. But what a treat to be able to see all of these very cool applications and people. I think having similar challenges, obviously, and being having a lot in common with each other, whether you're working in, you know, subterranean freshwater or marine water or soil mycorrhizae. It's a lot of commonalities. And so what we have on the schedule here for the next half hour, and it doesn't have to be that long. It's sort of as long as the conversation lasts, but a little bit of discussion as a group. It's sort of a nice size group. It's not too big, not too small. And so I've got a couple of general themes that I've written down that I think we can tee up here. And I guess the first one that occurs to me is about taxonomic annotation. That for a lot of people in their work, the results are going to depend a lot on whether you're annotating species individually or you've got OTUs and you're then putting them into a phylum and you're saying, okay, the OTUs of different phyla, they don't differ very much from one environment to another. And I think I put this in the chat for Juan Diego and at least one other person, very sort of similar challenges. So I thought I would ask the group, how are you doing that phylogenetic, that annotation with taxonomy? How are you putting names to your sequences? And do you have suggestions for people especially when there are not every species in the database, then what do you do in that case? And I thought I'd ask the group. I have opinions, but I want to hear from you.
So, well, I think this is one of the most challenges that I think we all have, for example. But regarding the question that Elizabeth asked me, I think Kraken 2 is the best option because we are working with MinION, so this data is really challenging to do it in chain or something like that, like the Silva database or Unite database for chain. It's really tricky to work with the ONT data. But currently we are using Kraken 2 and we have like great results. But I think the difficulty of Kraken 2 is the computational capability, because we need a lot of memory in the servers for these databases.
Great, thanks, Juan Diego. And Maja, you've got your hand up.
Yeah, I just wanted to say what I use because for me it's pretty much simple, 16S and 18S. So I use Silva and I use PR2, so the Roscoff version of protistal ribosomal database. And I actually use the modified primers for V4 region for 18S genes, so it's more compatible with PR2. So I get a really, really good results. But even though I have a lot of unclassified organisms, of course, in phytoplankton especially. But when I have a lot of taxa that are really abundant, speaking of amplicon sequence abundance, but I usually go and take those sequences by hand and check them out in BOLD, in GeneBank, in every other database that I know. Diet Barcode is great database for RBCL that I use often also. But yeah, that's a great problem with curation of those databases, because you need to have a scientist that is trained, that is taxonomist, that really curates those organisms, that looks at these organisms and really know what they are. So yeah, I didn't do yet koi, so mitochondrial genes, but I plan to for zooplankton. So if anyone has advice for that database, for which primers should I use for barcoding zooplankton, please tell me. Yeah.
Great, thank you. And Sophia has a hand. Before getting to Sophia, I want to highlight that those two answers, right? Maja focused on the database part of the problem, and Juan Diego focused on the algorithm part of the problem of how do you match a sequence to a name. And everyone on this call probably has both of those problems. And it depends. Yeah, maybe if it's easy in a sense, as Maja says, if you are using 16S and bacteria or whatever, there are these databases that already exist. For many of us, we don't have databases that already exist, and we're in the process of trying to create databases that are relevant to our own purposes. And so I think, yeah, a lot to talk about there. But I don't want to talk too much. Sophia has a hand.
So I hope I have understood the question. We're using ISVs, so each unique read sequence doesn't get grouped with another. And right now we have used like this identity thresholds. And I think we're in the process of trying to do this, so I'm not sure if I can explain it very well, but basically what we will do in terms of defining those similarity thresholds is that we have our the database, the sequence database for the species that we are targeting, and we're going to try to see like how much difference is, how much variability is within a species, within a genus. So then we can say if it's 98% similar, it should be the ISV should be attributed to a species level identification, or if it's 95%, it should be to the family or maybe the genus. So I think that would be like a good approach, but also takes a lot of work because it's according to the context of your study and the species that you are working with. Yeah, I hope I have explained that somewhat.
Great, thanks, Sophia. This is again, the chat is going on, and to repeat what we said before, we will be saving the chat and putting it into the Google Doc. So don't worry about trying to write things down. We will capture that for you, and you will have access to it going forward. Are there any other questions? We could talk about this topic all day long, and I'm happy to. But are there other questions maybe before we spend too much time on this one? Karianne, please.
Yes, so I saw in the chat that you advised against blocking primers, and I'm actually planning to use them because I have mollusk primers and then I have fish blocking primers to block the fish DNA. So I wouldn't know why to not use them, and I would be happy to learn.
Oh, of course, it's an opinion. It's not a law. Uh, yeah, sure. So this is a problem that many of us experience, where you have target species that you care about. Maybe if it's a diet study, for example, and you want to block the predator, and so there exist primers that you can design or buy that block the amplification of what they match, of the taxon they match. So it seems very attractive to say, I want to block all fish from amplifying, but I only want to amplify everything that's not fish or in the gut contents, for example. And this works. It's just that the blocking primers can work a little unpredictably, like all primers. So they will block, probably they will block your fish, but they may block other things too. Almost certainly they will block unpredictable things. So I think the way to do it, again, this is an opinion, but if we treat all primers like an assay, not like truth, right? If you design fish primers, they will amplify fish, but not all fish and not only fish, right? There's some uncertainty. And the same thing with blocking primers. They will block some things, but they will not block all things, and they will block some off-target things also. And so just treat it with a little bit of uncertainty, but the assay should work predictably, if as a comparative matter, right? From one sample to the next, you can compare those results and they will make sense. It's just that it's not absolutely 100% doing the thing you want it to do.
Oh, I was going to add, but let me see if Tessa wants to add first.
It's okay. You can add first.
Okay. I was just going to say to kind of expand a little bit on what Ryan was saying too. We have found a lot of times over and over again, what says it will work on the computer in silico is not exactly how it works in the lab. And so even if it looks like the blocking primer designed should only block the thing you're trying to, it might not. And so I think there's just an extra level of testing once you actually move from designing it perfectly on the computer to the lab. And so I think if your host is going to eat up all of your DNA sequences, that isn't helpful either. But it just requires an extra step of testing and really understanding what's happening with the blocking primer, just because the in silico results never quite match what happens when you actually put it in a PCR tube and send it away. So just advocating for, if you do use them, doing some testing before, so you really understand what is happening, and using like a known mock community or something beforehand, and then seeing what gets blocked and what doesn't get blocked.
So for example, I designed human blocking primers because I didn't want human sequences, and it's 100, I forget which gene, I'm not sure, a mitochondrial gene. And it was 100% matched to humans and nothing else. And it blocked fish also, and all kinds of other vertebrates. So that's, what is that, 100 million years of evolution, 150 million years. And so you just never know what it's going to do. So it's good to do, if you can have samples where you're treating them with blocking primers and without, and sequence both of them. That gives you confidence in what your blocking primers are really doing. Okay, Tessa, sorry.
Yeah, no worries. No, I just wanted to add that in the preliminary study I was talking about, we actually did half of the samples with a blocking primer and the other half without. And what was really interesting was the samples that had the blocking primer, the host DNA was like double or triple with a blocking primer, which didn't make any sense. And so we were like, all right, we're just going to continue without blocking primers. We didn't design them ourselves. We had a company where we send our samples to for sequencing. They designed the blocking primer and stuff for us. And they were like, you guys should definitely do a blocking primer. There's going to be a lot of host DNA. And we're like, okay, we'll do half with blocking, half without. And yeah, when we got the sequences back and everything, and how there was double or triple the host DNA, I was like, I can't believe, like, it was really nice to see that, because now we're gonna continue the study without blocking primers. But I'm not really sure what the explanation is for that, and I don't know if you guys, Ryan or Elizabeth, if you guys have ever seen that before or heard about it doubling host.
I love that it was the opposite. It was an anti-blocking primer. It was,
It's a promoter.
yeah.
It really
No,
was.
I've not heard of that specific problem, but yeah, anyway, this is just you never know what you're going to get out of the tube, and you just have to try and find out. But it's also one thing that's general, I think, to everyone on the screen that's worth knowing is when you get off-target amplification for species you don't care about or you don't want, it is okay to ignore those. You can say, I'm going to just take the species that I care about and analyze those, because it's not cheating to throw out species that you don't care about from your data, because the metabarcoding data is compositional. These are proportions, right? So you're looking at all the proportions, they have to sum to 1 in your sample. And so if you subtract out human or cow or other things you don't care about from your data, you can just renormalize the data, and then so that the proportions again sum to 1, but the proportions of the things you care about. And that's a totally acceptable analytical thing to do. It's not cheating. And so that's another solution, Kariana, to the problem. Instead of blocking them, you can just remove them bioinformatically later, unless they've taken up 90% of your reads, and most of your amplification is off target, and then yeah, that's bad.
Ryan, I'm going to add here too. I think this is a discussion point that I had written down that I thought might be interesting, and kind of going back to CO1 and the conversation in the chat with Chiara about how you can get really different answers each time you sequence with CO1. And so what you were just saying with subsetting species that you care about and renormalizing works really well when you also feel like you have reliable information. But with choosing a primer set with degeneracies, you have the benefit of you get a lot more species. You get a lot more range of species across the whole tree of life. But with Chiara's experience, it's also really sample to sample, and you might get a lot of variability. And so a degenerate primer set is not one primer set, it's many, many, many combinations of primer sets. And so it's less predictable. You don't know every time when you run that PCR cycle which primers of which degeneracies are going to get picked up in those early cycles of your PCR and then amplified over and over again. And so I guess just to talk to the group about and see what other people's experiences are, it's really helpful to get information on a really wide range of taxa. But what we have found is what I think Ryan put in the chat as well is there's also a really big benefit to knowing exactly what you're looking for and using primers, using multiple 2 or 3 primer sets to look at more targeted groups of organisms, rather than using a single primer to catch everything, just because of that unreliability and unpredictability and sort of the luck of the draw of what gets captured in those early cycles and then gets overrepresented or and shows up in your replicates. So I guess that was a long-winded way of saying, if there's anybody else, Chiara, I don't know if you want to chime in, or if anybody else has had a similar experience with CO1. I assume the Leray primers are the most commonly used ones where it's every 3rd base pair is a degeneracy. So they're, a 1/3 of them are variable. So yeah, go ahead, Chiara.
Yeah, thank you. I just wanted to add one thing. This was the first experiment, so I didn't know what to expect. So I said, okay, I got this. It was an old eDNA sample that I used to find a specific target fish. Then I said, okay, I had these samples already extracted, they are freezed. Why not to check what's in the wall in total, So that's very good in eDNA samples because you can go back and see the big picture instead of a single species. By the way, in my experience, we tried Lere primer, 350 base pair roughly, and the shorter CO1, I don't remember the name, 150. And with the smaller fragments, we got more, I mean, a list of species that was bigger. It makes sense because the amplicon was smaller. It makes sense. But the zoom on the picture was much, at least was bigger. I don't know if it was better. We are trying to understand. Was bigger than using the bigger fragments. So I saw in the chat someone saying, well, but using a bigger fragment, it should be, I mean, it gives you more information. You cannot go on a big fragment on eDNA. I mean, it's you can get it on DNA from tissue, for example, but not on the eDNA. And also, what I wanted to say is that reference database is also a huge problem. I mean, at the beginning, we tried to just take it out, taking all the sequences from the NCBI and working on the big database. Then realized that sometimes there was like, I don't know, misassignment or some errors. Something wasn't very good. So we selected sequences by certain query. For example, in my case was freshwater, but I wanted to exclude by downloading the sequencing, for example, spiders, because I know that spider doesn't have larvae, aquatic larvae, you know. So I pre-selected the sequences to construct, to build the reference database. Now we are doing that, so I cannot say anything about the result or how good it is. We are trying to understand. Thank you.
Carrie Ann, you want to go ahead and chime in too?
Yeah, I wanted to ask how you said you pre-select specific groups for your own reference database. How do you do it? Do you just say, oh, I only want mollusks, and I don't know, how do you do?
What we did was by selecting the query and use the advanced search at the gene bank, at the NCBI, we said we want all the sequences. They are named CO1, cytochrome, CO1, COE capital letter, for example, because, you know, they're written even in different ways. Then the full name, then COX1. So all the combination that to us were reliable as people can write CO1. And then we said CO1, let me think, and exclude specific group of animals. Like I said before, this was a freshwater, water, let's say for water in general, samples. So I can keep mosquitoes larvae because they have aquatic life during the first stages, but I excluded completely spiders, because if a bird flies on my rivers and lose their prey and there's a spider, I will find a sequence from a spider, but it's there just by chance, not because it's an animal living there. I don't know if it's clear or not. I can, if you want, I can send you the query that we have prepared. Yeah. And then you can tailor it and see what is better for you, or what you can exclude more or include.
If you want to share, it will be super nice. It will be super nice. You're very good.
This touches upon another question that is very common, I think, to many of us, which is, what is a false positive? And there's this interesting paper. I was just going, I'll find the link and put it in the chat. a guy named John Darling in the United States in a federal agency, environmental agency, has this paper called, and Adam Sepulveda, I think, is the other person. So two US people. It's called What Do You Mean by False Positive? And it can be this problem where the DNA is there. The DNA is truly present. That's a true finding, that the spider is present or whatever, Chiara. But maybe so the spider DNA is present, but the spider itself is not present in the sample, right? And is that a false positive? I think of that as, no, it's not a false positive. The DNA was truly there, and that's what you were surveying. But in another sense, it is a false positive if it's the animal you care about, the organism itself, right? And so this paper, thank you, Eiley, for she just posted the link to it. I think is a really useful framework for thinking about that. What do we mean when we say false positive? Because in one sense, yes, in another sense, no. And you can, when you're writing and when you're thinking about these problems, you can be specific about what sense you mean that phrase. I love that we have actual code happening in the chat. This is a first. This is great. And this is the, I missed part of that conversation, but it looks like maybe it's the query for GenBank, for the NCBI query to apparently be a very specific, exclude very specific things. Okay, I think we probably have time for one or two more points of discussion before Kate is going to wrap it up here for us. Any other questions? Now we're all just looking at the NCBI query code. Yeah, Maja, please.
Yeah, just a thought that is going on in my head over a few weeks ago. So when it's time to switch, not to switch, maybe to do both in parallel, and metabarcoding and metagenomics, especially for environments like marine environments, like water, like not host specific. So for me, that's the important question, because for metabarcoding, I'm just getting, I just feel like I'm getting just snips of the information. I need ecological function. I need metabolic interconnection of the organisms. So is it what do you think? It's normal to just switch to metagenomics, to shotgun sequencing and search for metagenomes and then analyze that, or to do both, and metabarcoding and metagenomics?
I love that your brain can handle all of that at once. It seems like I find it scary, daunting, but I'll put a link in the chat. We work in with a researcher here at the University of Washington, Ginger Armbrust, and her lab does exactly what you're talking about. So they do functional genomics out of, in her case, the ocean, but it doesn't matter what the environment is. And they view an ecosystem not as a collection of species, but as a collection of functions, gene functions. And so then you can look for the alternative. Instead of looking at shotgun sequencing and all of the genes of everything, you can look for functional genes and ask which functional genes are present. And then it's a simplified view of the ecosystem. But they also have a lot of computing power so that they can reconstruct genomes out of mixed template samples. So there are ways of doing that. A guy named Vaughn Iverson has the code for doing that, and that's all published. Oh, Eiley's got the link in there. So that's at least one way forward that people have thought about that. It's obviously a big adventure. I'm now just appreciating your headphones also, which are the coolest headphones I've ever seen.
Thank you.
Yeah. I think Aruj had a hand up next.
Yes, thank you. About the blast search at NCBI, and there is a lot going on in chat section. Actually, different groups have conserved region. For example, if we talk about the fungi, we know that the ITS region, ITS1, 5.8S and ITS2, this is conserved for the fungi. So if I found a species, a fungal species, whether in the form of fruiting body or in the form of morphotype, I will blast search at NCBI. If I get the value about 90, 91%, most probably it will be something new. Secondly, we have a unite analysis, UNITE, for the ectomycorrhizal root. There are some other database for fungi. So the value, threshold value 97, just give us the values of OTUs, Operation Taxonomic Unit. For the confirmation, I think in the case of fungi, we have to go for the multi-gene phylogenetics as well as the morphological and anatomical study. So I think this value just give us a first indication about its position, but just to make the things clear, we have to go for multi-genes. I mean the LSU region, RPB1, RPB2, TEF, so we can, it will be clear that what will be its position. I hope it will make some sense.
Yeah, great. Thank you, Aruj. And I think you're highlighting what some of this has gone in the chat, as you said, that depending on which taxonomic group you're interested in, there might be really different rules of annotation and which genes are useful, which sometimes you need a nuclear gene. Sometimes you need many genes to be able to put a name on genetic information you're getting from the world. So I think that, yeah, that's exactly right. And obviously different ways forward in the future might include single cell analysis as we get there, where you're able to look at many genes and you know that it comes from the same individual, from the same cell. I think we're most of us not in that world yet, but we may end up there. But yeah, being aware of those different approaches in different groups is really important, rather than just, saying, oh, 97% similar automatically means a species. And there's literature out there on that point. Oh wow, a lot going on in the chat. Nuo, last word before we turn the mic back over to Kate here.
Yes. Just to say thank you, because this question is really important. How percentage we can consider when we are trying to blast some species. Clearly, when you are in our situation like tropical waters, sometimes when you blast, you don't have, let's say, more than 19%. It's like 85, 86, sometimes 90, if you are lucky. But the question I would like to ask is, like what we are doing with microorganisms, using, let's say, the OTU0, is it possible to think about this possibility for, let's say, fish, for example? Thanks.
I'm not sure I understood the question. What is, I don't know, OTU0. Tell me about that.
OTU0 is what they call also the ISV, like amplicon sequence variants.
Yeah, so it's things that are identical or very nearly identical, rather than clustering across a larger set of sequences. Do I have that right?
Yeah.
Yeah. And the question is, does that work? We do that for fish. We do that for animals. And the question is, does it work for microorganisms?
For micrograms, I think it's worked very well. Because let's say I'm trying to do it in my future study, because I would like to compare, let's say, the OTU and also the ISV. We show that, let's say, we can have a kind of difference between the two approach.
Mm-hmm.
But I think that it depends also what type of sequencing you perform.
Yeah.
Long read or short read. Probably with long read, it may be very interesting to have good results. But for short read, I don't know.
Yeah, I was going to say the same thing, Noah, which was how are you actually getting the reads? Because if it's minion, you might not have if it's Minion versus MySeq and length and all that kind of things, the clustering versus the using the unique sequence like ASV approach might give you really different answers. So I think it's a combination of taxonomy, whether it's, fish or bacteria, how the sequences were generated, and then the algorithm used to process. So we don't do a lot of work with microorganisms, but if anybody else on the call does, certainly reach out to Noel and you guys can tell us what we need to know.
Okay, well, I think we should wrap it up here in the interest of time. But once again, we could be talking, really, we could do this for a long time. And we are in the weeds at this moment with all of you. And I love that there's this level of detail of interest and conversation. And so please, I think as we started out the time today saying, the point of this is to create these connections, and you have each other's contact information, you have our contact information. And so this is not really the end of a conversation, even though it's the end of a Zoom call. And I will turn it over to Kate to take us out here.
Yeah, I think Ailey, you were going to go over the upcoming events.
Yeah, thanks, Kate, and thanks, Ryan. In the same vein as Ryan was saying, the conversation should not end at the end of the Zoom call. We really want you to all get a chance to know each other and meet each other and use each other as resources. So we thought it might be helpful to tell you some upcoming events that we are aware of that are kind of happening conferences, either hybrid or virtual or in person. So this is a couple coming up in the next few months. This is also in the Google Doc. And what we would love is if you feel comfortable telling everybody if you're going. So actually, Noel, I think we didn't ask you, but we know that you're going to GeoBond, so we push your name there. But the idea was that if you want to meet other people in person, or if you're going to the same virtual conference, you can chat and make sure that you see each other's talks, or meet up in a breakout session, that kind of thing. So please use the Google Doc. Like Kate and Ryan have said, this Google Doc will continue. You will have access to it. We're going to copy the Zoom chat over here. So if you're going to any of these, please add your name if you would like to meet other people actually in person, in 3D. And then if you know of any other sessions, any other meetings or conferences coming up that you're attending, please put new conferences on there. We know most of these just from collaborators telling us they'll be attending. But if there's other meetings that you're planning on going to, just drop them in that same Google Doc and we'll put them on our website. And sometimes we tweet out, who's going to this meeting. So just one other way for you to connect with people in person. And yeah, I think that's it for me. And I will hand it off to, I think Kara goes next. We have a couple of tips of the day.
Yeah, and so I just wanted to remind everybody for a networking tip. Take some time when we're done with the talk and contact somebody that you've met today. That's a really great first step. And then also just a couple reminders that are not on here, but we do have the listserv that's available to everybody. You're all on it. I will put the email in the chat as well. I'm not good at typing and talking at the same time, so I'll do it when I'm done. But feel free to send any questions you have. That's everyone that's received microgrants or has been a visiting scholar or has gotten a grant from us. So there's about 120 of you on there. And that's also a really great way to network with each other. And then one last mention about the blog. We were chatting about it a little in the chat, but thank you for those of you who have already sent us blogs, and we do take double submissions. Again, if you want to talk about something else you're working on, that's great. And if you haven't submitted a blog, feel free, check it out on our website. We love to talk about what everybody's doing and put that out in the world. So thanks, everybody.
Great. And so in the interest of time, just a very short science tip of the day. I think that many of us want there to be some truth that we are capturing, and often we take an average of things to try and get closer to that truth. But just a reminder that there is information in the variability that you're seeing, and so don't lose that information, but capture it. Measure how much variability is in your samples across technical replicates, for example, and that way you have a much better sense of the difference between signal and noise in your data.